So I'm Maggie Patterson, and I'll be presenting on the phosphorylation of myocardial proteins of hibernating 13-line ground squirrels. As some background, hibernation is a natural survival strategy for winter months that many mammalian species use. However, there are different hibernation models. For example, larger mammals like bears undergo the typical model that we think of, where they undergo torpor, which is where they have decreased vital signs of they're not as active, they're usually sleeping, they have decreased body temperatures, breathing rates, and heart rates. However, smaller mammals, which are our subjects here, the 13-line ground squirrels, have torpor, so these same decreased vital signs that are interrupted by these intermittent bouts of arousal, which will just be abbreviated as IBA, where their vital signs go back to what is normal for summer periods about every 2 weeks or so for about 24 hours. And so this graph just gives a brief overview of the internal body temperature of a single squirrel. And so we see summer active, the temperatures are comparable to humans of about 37 or 38 degrees Celsius. And then late September, we start to fall into hibernation, where we see this first decrease in body temperature, followed by a quick rise up and then back down. And so you can see the pattern until about April, when they exit hibernation, where we have decreased internal body temperatures of about 4 degrees Celsius, followed by a quick increase back to normal, and that peak of which is IBA, and then back down into torpor. And so then there are 3 phases that we've been investigating. We have summer active, so summer period, torpor with the decreased vital signs, and then IBA, which closely mimics summer active. And then some numbers for vital signs. IBA is comparable to summer active. Body temperatures are about 35 to 38 degrees Celsius. Heart rates are very fast, of 350 to 400 beats per minute. And then breathing rates are over 40 breaths per minute. Because of these increased heart rates, you have a shorter filling time, and so the heart would have to not only have increased relaxation rates, but also a decrease in stiffness in order to be more compliant. And on the flip side, we have torpor, where body temperatures have dropped to below 5 degrees Celsius. Heart rates are much slower, of 5 to 10 beats per minute. And then breathing rates are under 1 breath per minute. And so now we have a longer filling time, and so to protect against overfilling of the heart and stretching it too much, there may be an increase in stiffness, which leads us to the 2 proteins of interest that I've investigated. So the first one is cardiac troponin I, which will be abbreviated as CTNI. It is the inhibitory subunit of troponin. It plays a role in calcium sensitivity, which will also regulate relaxation rates, and it's a regulatory molecule. And the second protein of interest is heat shock protein 27, which is abbreviated as HSP27. This can affect myocardial stiffness as well as protein aggregation. And so this is a general graph that we see for force and calcium concentrations. And when we phosphorylate a specific site in cardiac troponin I, specifically serine 2324, we see this rightward shift. And so the rightward shift decreases the calcium sensitivity, so it's allowing us to have an increased relaxation rate. And this increased relaxation rate allows the ventricles to fill, despite there being potentially a reduction in filling time, which we would see with increased heart rates. And so because of this phosphorylation of these 2 sites of serine 23 and 24 is hypothesized to occur during those 2 periods in which we have increased heart rates, which are summer active and IBA. And then another site within cardiac troponin I is serine 4345, which kind of acts against those previous sites. This is phosphorylated by PKC. And phosphorylation of these sites can decrease cross-bridge detachment rates and increases calcium sensitivity, whereas the other sites decrease calcium sensitivity. And so we would expect to see these sites phosphorylated during torpor, where we have those decreased heart rates. And our other protein of HSP27, this is also known as HSPB1. And phosphorylation of specifically, I believe it's serine 83, has been shown to reduce stiffness. And so a reduction in stiffness is beneficial for phases where we're increasing the contractility, we're having faster heart rates, which is again summer active in IBA. And so in order to analyze these proteins, we first have to separate them out and transfer them to blots. And so separation we do with SDS-Page gel electrophoresis. SDS page allows us to separate out smaller proteins such as myosin heavy chain, actin, troponin, and HSP27. And then using Western blot techniques, we transfer them to nitrocellulose membranes, we stain them with Phosphorylation, and then we can store them in negative 80 degrees Celsius until we can probe them later. This is an example of an SDS page gel stained with Kamasi blue, where we can see the protein bands separating based off of molecular weight. And then down the bottom, we see a nitrocellulose membrane stained with Ponsu S, and so each one of those red stains are the proteins. And then the antibody probing protocol is fairly similar for all three. This is an example of the serine 2324 protocol, where we block it for 60 minutes and we wash it. The primary antibody is placed overnight, and for this site, it was 1 to 1,000 dilution. We then wash it again the next day, add in the secondary antibody, a 1 to 5,000 dilution for 60 minutes. We wash it, and then for the serine 2324, we scan it with SpectraMax I3X, and then we can strip it and reprobe for total. So that way we're using the same blots to not only look for these specific sites, but then also total protein. And the exact same protocol for both HSP27 and then serine 43 for cardiac troponin I vary just according to the manufacturer of the antibodies, but they follow a similar outline, other than they use chemiluminescence, so we have to analyze them with the Lycor Odyssey FC. And so this is an example of the same blot for the serine 2324 phosphorylation sites. And so here we can see our controls were phosphorylated, and we had some of our samples phosphorylate. And then for total cTNI, we see all of the samples had protein, and so we compare those two ratios. So for data analysis, we use image J to get blots like are shown on the side there. We take the area of each and compare them from the phosphorylated to the total. We correct them with the control of the LV just to account for any differences with the membranes. And we use ANOVA and multiple comparison tests. And so this is a graph of the data for the serine 2324 sites. We see that Summer and IBA have similar groupings versus Torpor had a decreased phosphorylation. And so for these sites, we saw that there was significant seasonal effect with a p-value of 0.005. And the phosphorylated TNI to total TNI ratio for Torpor compared to Summer active was also significant in the fact that Torpor had less phosphorylation, and the p-value was 0.02. Same thing went when comparing Torpor to IBA. Torpor again had less phosphorylation with a p-value of 0.007. And since Summer active and IBA have similar heart rates and similar vital signs, we didn't expect to see any change, which was confirmed when we saw no significant change between the two ratios with a p-value of 0.812. And so then looking at the other site for cardiac troponin I, serine 43, this protocol still needs to be adapted. These are preliminary scans where we see the soleus is actually reacting and we have a little bit of sample, and then the total troponin still reacted well. Again, these are preliminary scans because some blots actually had no signal, and then other blots had non-specific binding where we were seeing lines for other proteins. And then for HSP27 phosphorylation, we saw no phosphorylation at the specific site, but then total HSP27 we only had for our controls. Our controls are rat LV on the left and then rat soleus on the right, which led us to investigate whether or not this antibody was specific for our species, and we discovered that HSP27, while it's highly conserved, it was more similar to mice than it was rats, and so this antibody may just not be specific enough for the squirrel species. And so then future directions, we're going to try and reprobe for HSP27 in the serine 43 sites for cardiac troponin I, tweak those protocols and or get new antibodies that are specific. Also investigate other sites of post-translational modifications on not only cardiac troponin I, but other myocardial proteins as well. We can also take a look at ProQ staining analysis, look at overall phosphorylation levels, and see if there's any change between them. And so why do we care? Why are we looking at squirrels? So hibernation conditions are similar to ischemia and reperfusion injury. And so when we have torpor with those decreased heart rates, we're not perfusing, we're not pumping blood as well. And so that would be associated with ischemia. But then when we switch back to IBA, we're seeing these increased heart rates, we're perfusing again, which would have reperfusion injury. But we're not seeing any damage to the squirrels as they undergo this multiple times during hibernation. And so they appear to have an inherent protection mechanism. And so this would be clinically applicable. So with organ transplantations, you could potentially increase the viability of an organ you can have it on ice for a longer period of time because you're protecting against ischemia and reperfusion injury. Or looking at heart failure and MI, are we able to kind of protect against the ischemia that occurs and then the later reperfusion injury? And then if we could also discover how to quickly change cardiac stiffness, you could rapidly intervene with drugs to treat conditions such as dilated cardiomyopathy, where if we could increase stiffness, we could potentially treat this disease. And so then special thanks to the Alma College IPHS department who's let me do this research for the past 3 years, specifically Dr. Karen Ball. And then we work closely with Wayne State University with Dr. Charles Chung, and then the University of Wisconsin has given us the tissues. Dr. Hannah Carey provided them to us. But thank you.